It's my pleasure to introduce Professor McGarry as our Jones Seminar speaker. Matt is an assistant professor of engineering at the Thayer School, and his research program has been dedicated for many years to improving diagnostic medicine by non-invasively imaging the mechanical properties of tissue. He's been in the field for close to 20 years, maybe over 20 years. So Matt obtained his bachelor's degree in mechanical engineering in 2006, went on to complete a master's degree program at the University of Canterbury in Christchurch, New Zealand in 2008. At that point, he came to Thayer, got his PhD, and then went on to do 2 postdoctoral fellowships, one at Thayer, one at Columbia University. Came back to Thayer in 2017, joined as a faculty, and during that time, he's made and continues to make some really important contributions to the field of magnetic resonance elastography, which we're going to hear about this afternoon. So let's welcome our speaker, and thanks, Matt, for talking.
All right, thanks, Scott, for the introduction. So as he said, I'm here to talk a bit about my work at Dartmouth on imaging mechanics of the brain using MRI. Brief outline of what I'm going to talk about today. First, why we're actually interested in the mechanics of the brain, so tissue mechanics and structure. Then the techniques that we use to measure these, which is known as magnetic resonance elastography. There'll be 3 kind of flavors that I'm going to talk about. First, viscoelastic MR elastography uses shear waves to image the shear modulus. Anisotropic MR elastography uses, there's 2 types of shear waves in an anisotropic material, and from these you can measure the shear modulus and anisotropy. And finally, at the lower frequencies of cardiac pulsation, poroelastic models come into play, and these use a poroelastic slow wave, where you can look at the shear modulus and hydraulic conductivity. And finally, we've got some schemes to use that for the prediction of glymphatic fluid. Basically, if you want to look at diagnosing disease under a microscope, you're looking at the morphology of the microstructural components. So you can see these examples from breast cancer on the side. You can see obvious changes in the microstructure. Microstructural changes and their effect on continuum properties are the basis of a lot of different imaging modalities. So the changes in the X-ray attenuation is imaged with X-ray and CT, the nuclear magnetic resonance environment in the MRI, ultrasound scatter and absorption, and electromagnetic properties for the NIR, microwave imaging and EIT that we've seen at Dartmouth. If you think of tissue as a composite elastic material, kind of like a fiberglass, the continuum properties are reflected as changes in the relative geometry and proportions of each of the macroscale components. So in elastography, we're looking at imaging the mechanical properties of tissue, hoping that gives us some sort of a window into the microstructural changes that are occurring in a disease. So it's not just that diseases affect mechanical properties. Mechanical properties also affect the development of brain tissue and development of diseases. So this is the field of mechanobiology, and I've picked out a couple of examples here that I find interesting. The first one is a study that shows that the stiffness of the microscale components of your brain increases with age, and the behavior of particular types of brain cells is influenced by the stiffness that they grow on. Now, if you take older brain cells and start growing them on a younger, softer scaffold, they start behaving much more like younger brain cells. So how your brain cells behave is affected by the mechanical properties of their local environment. This other one shows that as neurons are developing, in the absence of chemical signaling, they will move towards the softer side of a stiffness gradient. So then this shows that the stiffness and the stiffness distribution is very important in the developing brain. So if we want to talk about how we're going to image stiffness, I'm a mechanical engineer by training, so I always like to start with like a simple beam problem. So this is a 2 soft materials sandwiched in between a stiffer material. So to do this, we'd apply a stress to the outside and stretch it a bit. Then by measuring the strain and knowing the stress, we can calculate the effective distribution of the elastic modulus here. The issue here for something like this in brain is that your brain is inside a skull. People generally like to keep their brains inside the skull and won't let you go and measure the stress, so we don't know the stress. The other option, so if you want to think about using waves to probe things, you can look at steady state vibrations. So for steady state motion at a frequency of omega, this is the equation of motion. The important parameters are down here on the table. And then you'll see some images of the complex valued shear modulus. So the real component of that describes energy storage, and the imaginary component describes energy losses or attenuation. It means basically that there's a phase lag between the applied stress and applied strain at a frequency. You can also translate that into some a different way of parameterizing it as the shear stiffness, which is basically the equivalent to the wave speed in a damp material, and the damping ratio, which is the standard engineering damping ratio, telling you how much energy is lost per cycle. Where a damping ratio of 1 would mean it would decay without oscillation, so you'd push it and it would just kind of ooze back without bouncing. This equation has the left-hand side of it has two different terms, and they kind of govern the propagation of two different types of waves. So the one on the left there in green is the shear wave term, and then the one on the red, is the compression or longitudinal wave term. So what these waves actually look like is shown here. So the shear wave is obviously a transverse wave propagating along this beam analogy, and then the compression wave is actually exactly equivalent to a sound wave. Now, if we look at if we've got a reasonable vibration in tissue that we can apply to brain, it's 50 hertz. If we look at the mechanical properties of brain and the wavelengths of these, the shear wave is about 30 millimeters. So, it's like a small portion of your brain, whereas the compressional wave is on the order of 25 meters. So we're really interested in the shear wave in this case. One application where you may have heard of shear waves and compression waves before is the location of the epicenter of an earthquake. So this is Christchurch, New Zealand in 2011, and I was working in the little engineering school over here. And the way that they determine where the epicenter of it is by measuring the arrival time of the shear wave and the compression wave. And if you know those wave propagation speeds, you know how far away that is. And if you measure that in a few different places, you can then triangulate where the actual epicenter was, which was just over there. Now, if we look at these shear waves in this beam analogy again, you can see this is a purely soft beam, has this kind of shear wavelength, and a stiffer beam has a longer shear wavelength. And if you look at a composite beam, you can have a shorter wavelength in the softer part and a longer wavelength in the stiffer part. So the wavelength is determined. This wavelength is determined by the shear modulus alone, and you don't need to measure the stress. So we can do this in brain. So if you want to take these type of wave measurements in actual brains, we use this technique called MR elastography. So basically, in this case, we apply a vibration source to the back of the brain. So it's pretty light. Feels kind of like you're leaning on a speaker. The frequency range can range anywhere between 25 to 1200 hertz, but 50 hertz is really the sweet spot and where we get measurable wave amplitudes and not too much attenuation, so they actually reach the center of the brain. So these are the sort of wave fields that we measure at 50 hertz. So we've got three-dimensional wave fields through the full brain. And these are the fastest scans will get these at about 2 millimeters resolution in around about 2 minutes. Then finally, once we've taken these measurements, we use inversion algorithms to figure out what the underlying mechanical properties are. So the actual inversion algorithm that I work on is called nonlinear inversion. So in this case, we take these 3D motion fields and then we get the geometry from the anatomical MRI scans and build a computational model. In this case, it's a finite element model, and then we can apply any sort of mechanical model to this to be the governing equation. Because these models are really big, they're on the order of like 200,000 nodes. So you're trying to invert to solve it, you're trying to invert a sort of million by million matrix. We break it up into a grid of overlapping subzones because we've got data everywhere. We can describe boundary conditions. We send each of those subzones off to its own separate processor on a high performance computing environment. And then we just do a standard optimization loop. So we start with a guess of the mechanical properties. Then we solve the forward problem or the finite element solution to get the estimate from the computational models. Then we iteratively update this estimate of the mechanical properties to minimize the difference between predictions from the model and the measurements. And we repeat this a number of times, and because we're on these little subzones, we then need to rebuild the solution as the union of the subzone properties. Then we go and randomize it again, break it up into a whole new set of subzones, and then keep on going around this process until the property estimate stabilizes. So these are kind of examples of the images that we get in brain. So this is the real shear modulus and imaginary shear modulus, or the storage and loss, the parameters that describe those factors. And we can also parameterize these into different stiffness maps applied. So this is shear stiffness and damping ratio, and you can put them on different color maps. We can look at repeatability, so you know that the same brain measured it multiple times in a row should be around about the same. So that's one way of knowing that we're right. But the real question that I've spent a lot of my career determining is, how do we know what the correct answer is? It's very difficult to, even if you can remove the brain from the head that you can in animal models, it's really difficult to actually do mechanical testing on it. And there's all these different inversion algorithms. So this is just an example. I've written down some of the names off the top of my head, but there's at least 10 different inversion algorithms around. And it's very tempting to just get these answers out that say that there's a stiffness and then just start using them clinically without really ensuring that you've got the right answer. So we go through a pretty intensive validation protocol for each of our inversion techniques that we develop. We start with a simulation where we prescribe known mechanical properties and we know exactly what the governing mechanical model is. So this is a really idealized case. We then simulate displacements from the finite element model. So in this case, we've just taken a bunch of brain structures and got properties from the literature. And then we've simulated what the displacements would be if you put them in an MRI or MR elastography-like environment. And then we pretend that we don't know the properties and see if our inversions can recover the true properties. And so in this case, we do a pretty good job, especially in the real shear modulus. We can also make it a little bit more realistic by adding some simulated measurement noise. So this is kind of on the higher end of noise that we would expect in MRI. So we're pretty confident in our estimates of the real shear modulus and moderately confident in our estimates of the imaginary shear modulus. So once we know that it works in this really idealized environment, we then do a bit more of a experimental environment and controlled phantoms. So in this case, we'll build phantoms with known stiffness and damping variation out of silicon gels, tofus, and gelatin, apply vibration and image using MRE, and then compare the stiffness estimates to what we know about the phantom materials. So either we just know which ones are stiffer and softer, and we know where we know the shapes of the inclusions that we put in there, and you can also do mechanical testing on controlled, in this more controlled environment. So this is an example that one of our grad students did a really good job on. So this is a silicon gel phantom, and we've got about 30 different inclusions here of different contrast. And then she managed to show that we can see an inclusion of only 14% contrast at 8 millimeters. And we did the same thing with damping ratio. So these are a tofu and gelatin phantom. You'll notice that these images don't look as clear as like typical some of the medical images you'll see, like MRIs. The main explanation for that is that we're trying to image sub-wavelength. So our wavelength's on the order of 30 millimeters, and we're looking for things on the order of 8 millimeters. So it's kind of a near-field versus far-field problem. It's a lot more difficult. That's why we kind of need to go into these like more complex finite element-based model fitting approaches. One of the first applications that we tried once we got this viscoelastic MRE validated and we were confident with our results is what we call the elastome. So you have a genome, a connectome, a biome. This is what we call the elastome. So we took 134 subjects, applied an MRE scan to them, and then registered them to the same atlas. So we can build these average property maps of the brain. And then this is publicly available, so anyone who needs these for any sort of science or any sort of modeling. And then we can extract estimates of properties of individual brain structures and their population variations. So here's some estimates of sort of subcortical grey matter structures and white matter. The next thing that we tried is which I think is really interesting, is structure-function relations. So in brain development, as you use neurons, they're reinforced, and ones that you don't use are pruned. So you end up with like just the ones that are being used. And then highly connected networks of neurons are associated with higher performance, although this isn't actually that clear because it's really difficult to, again, do these experiments, because to test the function of someone's brain, they need to be alive, and to look at the neuronal connections, they need to be not alive. So what we looked at is, do the mechanical properties reflect how well a brain structure functions? So if you think of this sort of mechanical composite analogy again, this is saying if there's more of a dense network of neuronal connections, can we see this in the stiffness or damping ratio properties? So to do this, we first looked at the hippocampus memory relationship. So if you've ever seen a TV show where someone gets amnesia, that's because they've had damage to their hippocampus. So it's very, very heavily involved in formation of memories. So the standard test to test your hippocampal function is this spatial reconstruction task. So we start with, we show the subject a set of objects, and then take them away for a little while, and then put them back up, and then tell them to put them back where they saw them before. And then we quantify the errors in that. So there's two types of errors: a distance error, where they put it in slightly the wrong place, and arrangement, where they switch to around. This is highly sensitive to the health of the hippocampus in patients, and it's also, as your hippocampus degrades, your performance in this test also degrades. So I was actually not all that confident that this was ever going to work. It seemed like a bit of a stretch, but we did actually find a really strong relationship between the damping ratio of the hippocampus and the spatial reconstruction task. And then that was not reflected in the volume, which is kind of the standard measure of hippocampal health, and then also the stiffness. Just because the stiffness isn't affected, there's other compensatory mechanisms. Your brain has to do more than think. It also has to stay in the right shape and respond to impacts. So it doesn't really want to create huge changes in stiffness, because that creates stress concentrations. So we've actually, so the gold standard in these sort of structure function tests is called a double dissociation. So that's when you take two different structures which have different associated functions, and then you show that whatever you're measuring is sensitive to the function of a particular structure, but not sensitive to the other function. So this is the hippocampus and orbitofrontal cortex, and we showed that the damping ratio of the hippocampus is strongly related to the memory task, but not the fluid intelligence task, whereas the fluid intelligence is associated with the orbitofrontal cortex, and we detected a significant relationship between performance and fluid intelligence in the orbitofrontal cortex, but not in the hippocampus. So we've done this in a few other things. So we repeated the first hippocampus study in a larger sample. We've tested the hippocampus and episodic memory in particular in older adults, and we've found these same relationships. And then we've also done another double dissociation of the hippocampus memory and a different structure called the ventromedial prefrontal cortex, which is associated with rule learning. So because we've done a really careful job of ensuring that we've got spatial and quantitative accuracy, we felt confident moving right down to the very small parts, the subfields of the hippocampus, which are associated with slightly different types of memory. So in this case, we used our highest resolution protocol, and then we found that verbal and relational memory were most significantly related to the CA1, CA2 region, and then verbal recall was predicted by the dentate gyrus and the CA3 region. So this shows that different parts of memory are associated with the parts of the hippocampus which are understood to be associated with that. And then finally, we also looked at these hippocampal subfields in an early stage of dementia, which is called amnestic mild cognitive impairment. So this is kind of like, you can think of this as a pre-Alzheimer's disease. And what we found is that these same regions were also preferentially affected in the early stages of Alzheimer's disease. So this suggests that the same processes that make sort of you and I have different memory, some people have better memories than others, those same processes are happening in diseases which affect memory. And then we also showed in the study that using MRI on top of volumetric measurements improves the diagnostic accuracy. Some other interesting studies we've done with viscoelastic MRI is we've detected mechanical signatures in Alzheimer's disease in the cerebral cortex. We've seen mechanical signatures of reward and control systems for adolescent risk-taking, dynamic balance reactions in children with cerebral palsy. And then finally, we've really started moving into animal models where we can sort of make changes and validate them through histology. So this is a recent study that we've just finished in a rat model of fetal alcohol spectrum, and showing that exercise can recover some of the deficits from early alcohol exposure. So all these things now, these hippocampus and most of the stuff we've been looking at has been in grey matter structures. Grey matter is particularly appropriate for isotropic viscoelasticity, which is that model. But a lot of brain is white matter, which has aligned tracts of axons, so it's not really isotropic. So the stiffness is actually stiffer along the axonal tract and softer across it. So we come up with this little model, and we think that by measuring the anisotropic mechanical properties, we can separate different disease processes. So neuroinflammation involves just recruitment of a whole lot of immune cells and a lot of fluid, and then we think that this should reduce the average stiffness, whereas demyelinating processes like multiple sclerosis preferentially attack these stiff myelin neurons, so we think that will reduce the anisotropy. So we can come up with a mechanical model for white matter. You can use this another MRI technique called diffusion tensor imaging, and this tells you where all the fibers are and which direction they are. So if you know what direction the preferential, what direction is the axis of symmetry or the stiffer direction, you can make a pretty big simplification to the full anisotropic elasticity problem, and you can get this nice symmetry. We use a fiber-based property description, where we've got a base shear modulus, which is kind of like an average shear modulus. We've still got a damping ratio. And then we've got two types of anisotropy. The shear anisotropy, which tells you how much stiffer the material is in shear along the fiber axis and planes involving the fiber axis. And the tensile anisotropy, which tells you how much the tensile stiffness changes along the fiber axis versus across. Fibrous material has two different types of shear waves, depending on the polarization direction with respect to the fiber angle and the propagation direction. You've got a fast shear wave and a slow shear wave. So if you measure a brain vibrating and both these types of waves are present, you should be able to compute the stiffness and the anisotropy. The actual algorithm that we do is based on the nonlinear inversion finite element based thing I showed you before. The inputs are the brain geometry, so we create the finite element model, two different displacement fields, so we get a bigger diversity of wave propagation directions. So we vibrate from the back of the head in the anterior-posterior direction, and we do a separate experiment where we're vibrating from the left and right direction. So there's two different displacement fields where waves are bouncing around everywhere. And we also know the fiber directions from diffusion tensor imaging, and then the output is these maps of the shear modulus, you see my, yeah, you can see my cursor. Shear modulus, the shear anisotropy, the tensile anisotropy, and then also the damping ratio. We went through a similar, very careful validation process here, where we started with a simple simulation of a shell. So this is kind of like a shell with fibers going tangent to the shell surface and pointing out. So you can think of that as like the lines on a watermelon. And we made sure that we could independently recover a variation in each of these properties by themselves without too much of an effect. So you can see there's a little bit of ghosting of or crosstalk as we call it, but in general we do a pretty good job of recovering the true mechanical properties. Then we've got a more realistic brain simulation where we apply just kind of some properties from the literature to a brain-like geometry and apply a realistic set of boundary conditions, and you can see that we've got the true values here that we prescribed in the simulation, and we pretended we didn't know them, and then showed that we can recover each of these properties pretty well all at once. We've also done a 3D printed anisotropic phantom, so we printed this lattice in this vintile pattern, and it's stretched in one direction, so it's not an isotropic lattice, it's kind of elongated in one direction, and that's the direction of anisotropy. We embedded this in a kind of a gelatin matrix, so we expect that in the circle here, yeah, you can see that. So where the lattice is, we expect that to be anisotropic, and then outside to be isotropic. And we do a pretty good job of recovering the. Sorry, I forgot to put the labels on this, but this one here is the red one here is the shear anisotropy, and this one is the tensile anisotropy. And then finally, we can obviously apply it to in vivo brain, but we can't really, without knowing what the mechanical properties are in advance, we're not sure that it's right. But by doing this careful simulation and more idealized, or careful validation in a more idealized case, we can be more confident of our results in brain. This is kind of newer stuff, but we have got into the first few clinical applications. So we've published the first set of anisotropic mechanical properties of a whole lot of different white matter structures in human brains. We've started doing experiments in pig models, looking at brain development in the mini pig. And then we've also done, obviously, these kind of general imaging techniques in a single a fibrous tissue is also muscles. So we've done 2 studies in muscle using the gastrocnemius and the forearm. So figuring out how each of your forearm muscles engage using stiffness measurements. So there's not just a single fiber direction in some cases. The brain is obviously a really complex organ, and then there's all these different tracks going around, and sometimes they'll cross over each other, sometimes they'll branch out. So up to 30 to 90% of white matter has been estimated to have complex fiber configurations that could be 2 or more fiber directions. And this causes a model data mismatch for our single fiber algorithms. And the question that we asked is, can we use an even more sophisticated mechanical model? So I always believe in models should be as simple as possible, but not too simple. So you want to kind of push things until they break. So this is the most complicated that we could think of. So we thought we'd give it a shot and then see what the limit is. So to do this, we needed to build a mechanical model for a 2-fiber system. So we're lucky we had a really good postdoc from the physics department. Damian Sawinski, he developed this 2-fiber model, but starting from sort of first principles as a physicist. So you take the hyperelastic potential and then you decompose it with a bunch of the invariants and pseudo-invariants based on fiber directions. And then this is a lot of math, but the basic thing that it boils down to is you allow anything that is not explicitly not allowed is allowed, and after that we end up with 7 terms. And each of these has a modulus associated with it. These are kind of a little bit abstract, but they can easily be mapped to like the properties of the fiber. So the average shear modulus and then the anisotropies of each of the fibers. A 2-fiber material still has just 2 types of shear waves, the fast and the slow wave, but they're a function of both fiber properties now. And we can actually, multi-shell diffusion tensor imaging can tell us both of these fiber directions in advance, so we can compute this model. The really cool thing about this model, from my perspective, as the guy who actually codes it, is that it's hierarchical. So we can add extra complexity to a material by just adding more terms. So if we only include 1 term in this big sum of terms below, that gives us the isotropic model from earlier. If we include 3 terms, we can have the single fiber anisotropic model, and then adding a few more terms gives us the 2-fiber. So we can build all these in the same subroutine as just like an extension. We used a similar shell phantom to the single fiber one, but in this case we had 2 fibers. So we have fibers going tangent to the shell surface pointing up like the watermelon, and then another set of fibers going straight in the x-axis. So this gives us every single possible interaction, every single possible intersection angle of fibers in a single simulation. Then we simulated vibration of this in 2 different orientations, just like we can with brain for AP and MR, or AP anterior, posterior, and left, right. And then we looked at, can we recover contrast in just one property? So these are reasonably busy plots, but each one of these is 8 different materials. The truth that we applied to it is 8 different materials with contrast in only 1 parameter. And then we've got 8 different parameters. So we've got the 7 moduli plus also 1 parameter describing damping. And then this is what we recovered. So we do a really good job for the first 5. There is a little bit of crosstalk. You can barely see that on that screen, but the interaction moduli are pretty difficult to calculate, so we ended up just lumping those into 1 parameter. But this shows that we can do reasonably well in a very idealized case. Then we tried this with a single in vivo case. So we took real MRI data in a brain, and then we did the isotropic inversion from the first flavor that I talked about, then the single fiber inversion and the 2-fiber inversion. And what was really surprising to me is that the images, one, that we were able to do this, so it really is a lot of parameters to be extracting from the data. But the decreased model data mismatch from better mechanical modeling on these crossing fibers actually made the shear anisotropy images look much better and much more realistic in terms of brain anatomy. We weren't really able to recover either. This is really small or really difficult to recover. So this is the interaction moduli, but we can get so from a single exam, we can get the average stiffness, the damping ratio, and then the shear and tensile anisotropies of 2 different fiber families from a single exam. This is very new stuff, but the actual, because here we're reconstructing 2 fibers everywhere, there's a lot of places where there isn't 2 fibers, so it makes it really hard to interpret the results. So what we want to do in the future is do a multi-model reconstruction. So we're using this kind of hierarchical 2-fiber model where we truncate, where we throw away some terms where they're not necessary. So any grey matter regions will just have 0 fibers or isotropic. Anywhere where there's only a single fiber family detected, we use the 1-fiber model, and then use 2 fibers only where it's necessary. So this controls the number of unknowns and kind of stabilizes the whole problem. So up until now, we've been doing external vibration, but the structure of the brain gives you another option. You've got a lot of blood being forced into your brain, and it's like at a reasonably high pressure of 6 kilopascals from systole to diastole. And then you've got this big network of sort of tubes in there, and then they get pressurized, and this actually causes a pulsation in the brain. So we can actually measure that pulsation using similar techniques to elastography. So these are retrospectively gated motion-sensitive sequences. So we just take a normal, usually just a normal, any sort of motion measuring thing. So there's a lot of angiography or cardiac sequences. And then we just make these sensitized to really slow motions. And these are the sort of motion fields we get. So here you've got this is in the left-right direction. So this is sort of a stretching this way. And then in the anterior-posterior direction, you've got a stretching this way. So you can see the front's moving forward and the back's moving back. And then in the foot-head direction, your brain's kind of going up and down like this. And you can see that in this animation here. At these lower frequencies, poroelasticity becomes more relevant. So the basic model of a poroelastic material is a fluid-saturated sponge, as you can see by my very realistic illustration here. The relevant properties for a poroelastic material are the elastic properties of the matrix, so similar to the isotropic viscoelastic material. So the shear modulus is the important one here, and then also hydrodynamic properties, so how the fluid component of this poroelastic model is interacting with the porous matrix. So the terms here are the porosity and the hydraulic conductivity. The hydraulic conductivity is very interesting because it's the proportionality constant for Darcy's law, which tells you how much of a flow you'll get given a pressure gradient. Poroelastic equations are here below. They look very similar to the viscoelastic equations with just some extra terms. But then, so the interesting thing about poroelasticity is the slow wave. So the slow wave is actually, if you've ever heard of an arterial pulse wave, it's when you force an incompressible fluid into a elastic, anything that's elastic, it kind of bulges out and that travels as a wave. And the wave of that is determined by fluid properties and also the sort of resistance to deformation or the stiffness of the of what you're pushing it into. So then the poroelastic slow wave is kind of the continuum limit of the arterial pulse wave. Because we're at 1 hertz, the shear wave doesn't really work anymore. Because of the stiffness of brain, the wavelength is on the order of like 1.7 meters. So you only get a tiny little part of the wavelength. So this is a very realistic rectangular brain. You can't see any wave propagation. It just looks like you're kind of squashing it really slowly. So we can't really get any estimates of the wavelength or get the properties out. But the poroelastic shear wave the poroelastic slow wave on the other hand is much lower speed. So this is a poroelastic material of the same shape, and you can really see the wave propagation. So we can get an estimate of the wave speed. And then the wave speed is a function of both the stiffness and the hydraulic conductivity. So we should better recover that. Similar sort of validation process for poroelastic imaging. So we have the simulation here where you've got contrast in the stiffness, positive, 0, and negative in the horizontal direction, and positive, 0, and negative contrast in the hydraulic conductivity in the vertical direction. So this gives you all possible combinations. You can see that we do a pretty good job of recovering the shear modulus and a reasonable, okay job of recovering the hydraulic conductivity. But the hydraulic conductivity is really hard to measure. There's no other ways of doing it, so this is still valuable. We tested this in a tofu and gelatin phantom where we did a gel in a tofu background and tofu in a gelic background. We expect tofu to have a reasonable amount of fluid mobility. If you've ever cut up tofu on your bench, fluid comes out of it, and gelatin, nothing comes out of it. So we see we've got the correct contrast and shear modulus and also hydraulic conductivity. But again, the quality of the images are a bit worse, but the difficulty of measuring that parameter is a lot higher. And then we did this in vivo. So one of our grad students, Lidgen Solomon, helped us out with this poroelastic hydrocephalus study. So hydrocephalus is known as water on the brain. It's a disorder in the way that your brain reabsorbs cerebrospinal fluid, and what you get is these really huge ventricles that are very obvious on an MRI scan. The problem in particular that they're interested in is that there is a treatment for this. So you can do a sort of neurosurgery process where you put in a shunt which drains some of the excess pressure off. Some patients respond really well to this, and then some patients don't. So there's not really any sort of reliable method at this stage to predict which ones do, and obviously you don't want to do brain surgery on someone who doesn't really need it. The results of the study showed that we did see significant differences in shear modulus and hydraulic conductivity for patients with hydrocephalus-like symptoms, as in large ventricles and healthy controls. That's interesting that this is the first time anyone's ever detected contrast in the hydraulic conductivity between a disease state and non-disease state, but it's not all that useful because it's also very obvious that there's something going on with these people just from the size of their ventricles. Unfortunately, we didn't detect significant differences in baseline MRE properties, so we weren't able to show that we can predict which ones respond and which ones don't, but we did show that imaging over time shows a interesting time course. So the stiffness, the evolution of the stiffness and hydraulic conductivity is different for people who respond and don't respond. So that gives us some interesting data for designing future studies. Finally, because we have these pulsatile displacement measurements, the pulsatility of brain is actually really important in a process called the glymphatic system, which is how the brain kind of flushes out waste. And that's important. So when these waste products, when there's dysfunctions in this, you get buildup of waste products, and then it's hypothesized that that causes diseases such as Alzheimer's disease or chronic traumatic encephalopathy in football players. So if you're taking a lot of knocks to the head, it kind of disrupts the way that fluid moves around your brain, leads to a buildup of bad proteins. And one of our new collaborators actually has a paper showing that the pulsatile component of blood flow is really important in the movement of fluid through this glymphatic system. So you can see these are small little microspheres. This is a mouse with a little window in its head in 2-photon microscopy, and you can see that the pulsatile movement of these microspheres in the interstitial environment is perfectly synchronized with cardiac pulsations. So that's kind of the driving force for moving these waste products to the brain. So can we measure that using our sort of MR elastography techniques? So the poroelastic model has a fluid source term as a driving force of the fluid pressure equation. So this is a very simple simulation where you've got a fluid source in the bottom corner of this cube, and you can see it's just pulling it in and out as you force fluid in and out over time. And these actually look really similar to the actual pulsations that we measure in brain. So you've got no muscles or anything in your brain, so the only thing that's producing this pulsation is fluid being forced in and out. We can measure that using similar techniques to MR elastography. So MR elastography, where this is the finite element system, we're estimating the mechanical properties which are inside the stiffness matrix. It's not too much different to estimate the forcing vector here. The forcing vector for the stress equations tells you the boundary conditions and body forces, which are approximately 0. But the interesting ones are the fluid pressure equations, where the internal nodes, the forcing on that is this internal fluid source. We can recover this with a similar optimization type process called generalized least squares. So we assume the properties are known. We could measure those with elastography, obviously. And then we recover a forcing vector which minimizes one term, which keeps the size of this forcing under control, and then also the difference between the displacements and the predicted and measured displacements. There's some weighting matrices in here, which control which terms to recover. So basically this tells you which one of these right-hand side terms. So we just set a really small weighting, or we set a high penalty for terms that we don't want to estimate or we know that are supposed to be 0, and a low weighting for low penalty for the internal fluid source. So if we look at what happens when we apply this to a pulsatile MRE data set, this is kind of what the fitted model looks like. So it's obviously much smoother, but we do a very good job of assimilating the measurements into our computational model. The fluid source and fluid pressure maps look like this. You've got a real and imaginary component because these are harmonic over time, so they have an amplitude and a phase lag. It's kind of hard to decipher what you're really looking at here, but if you look at it in terms of orderless slices, so this is a montage where at the top of the image is the base of the brain, and as we move through, you're moving towards the top. So as this blood pulse comes into your arteries here, it propagates through the brain and up into the upper slices, and we can measure the time variation of this. So if we want to talk about how this pulsatile motion forces glymphatic fluid around, we can go back to our model of the glymphatic system. So in this model, CSF or fluid, glymphatic fluid flows down the periarterial spaces, so the spaces around the arteries, and then gradually oozes its way through the tissue and then back and drains back out spaces along the veins. So what we do is we lump all of this kind of stuff in the middle, and then we call that the solid part of our poroelastic model, where this is the porous solid kind of soaked in a very slightly mobile glymphatic fluid. So the issue with trying to measure glymphatic fluid motion is it's really, really, really slow, like on the order of a few centimeters a day. So the only way to do it is to give people a contrast agent, like inject it directly into the brain or into some local area, and then come back and image them over the course of a few days and look how far it moved. So what we're doing is, rather than directly imaging the movement of the fluid, we're imaging the force, the driving sources or the forcing that drives it. So in this model, there's these blood vessels would be a kind of a continuum network of blood vessels, and this is an active fluid. So the fluid sources we were calculating before, we consider that to be blood. And what they do is each time the blood vessels filled with fluid, it applies a stress on this porous matrix of glymphatic fluid, and then moves it ever so slightly. And the factors which produce the pressure gradients are the stress in the solid matrix. And the good thing is, because we've assimilated these displacements into this computational model, we can compute things that we can't directly image. So we can compute these stresses in the solid matrix. So we get these tensile stresses and shear stresses. And then we've also got the pressure gradients of blood. And these interact through the equations of multi-compartment poroelasticity. This is a big mess, but you will note, if you look really carefully, we can actually see that we actually know the values for a lot of these through either elastography or just through knowledge of brain microstructure. And then the ones that we're imaging in this new technique here are the solid stress and the blood pressure gradients. And the only thing that's left over is this gradient of the interstitial fluid pressure, and that's the pressure gradients which drive glymphatic fluid. So this kind of forms what we want to do for the future directions of the lab. This is really new stuff, but we're really excited about it because this is obviously, glymphatic flow is not measurable, but being able to measure the driving forces for it is going to prove quite valuable. So summary, just in case you came here just for the cookies, measuring mechanics requires mechanical waves. There's three types of things that I've looked at. Isotropic viscoelasticity, where the shear wave determines the shear modulus. Anisotropic fibrous materials, you have a slow and a fast shear wave, and from that you can calculate the anisotropic stiffness. And at the frequencies of cardiac pulsations, the poroelastic slow wave allows us to measure the shear modulus and hydraulic conductivity. And then finally, with MR elastography, we just measure the propagation of these various types of waves and fit a computational model to recover the images of the governing properties. I'd like to acknowledge, obviously, I haven't done all this myself. We've had we have the MR elastography lab. We've had some really good postdocs and grad students, and then a lot of people that are giving me a lot of wise advice. And we've got a big network of collaborators around the world for these the more clinical and applied aspects. So thanks very much for listening. I will take any questions.